In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, how are you? I hope you all are enjoying your studies and taking rest and enjoying your test also and studying keenly. So this is our second spell biology class. As you know that it's a long time that we have um, just stopped studying biology. So this is our second spell first class, but overall lecture number 19. Dear students, I welcome you once again in Pakistan International School THAI's virtual learning system for the session 2020 and 21. So this is biology class for grade 8 students. Now you are set as ninth junior because you are studying all the subjects of ninth class. So once again we are here for studying our life of plants and animals. As you know that biology means the study of life. So we have finished chapter 1, 2, and 3 in the previous midterm. So now we are going to start from chapter number 4. That is very important chapter regarding this is said to be the basics of biology. Because before the discovery of cells, so the concept of biology is not known yet. So we will start today chapter number 4, that is about cell and tissues. So first of all, a very important topic which we are going to study here, that is microscopy and emergence of cell theory. Like discovery of cell has been started with the discovery of microscope. Because a cell cannot be seen without microscope. Microscopy is the study of microscope. So we will see that what type of microscopes are used and how they are discovered and formed, and then we will continue our topic, emergence of cell theory. So let's get started. We are on chapter number 4, that is about cell and tissues. So we know that all organisms are composed of cells. Some are composed of single cells and some are composed of many cells. Single cells examples you have studied about like bacteria, and many cells like human beings, they are called unicellular and multicellular. So cells or study of the cells is possible when we see them under the microscope. So the study of microscope is called microscopy, and all the processes of related to the microscope is called microscopy. So the use of microscope is known as microscopy. So the first compound microscope was developed by Zacharias Janssen. Is Zacharias Janssen, and this is its tube-like. This is tube-like microscope. You can see the emergence of light microscope, that is also said to be compound microscope. And this compound microscope you can see in your labs also, biology labs also, like it is having two pieces, eyepiece, and here you can have its objective lens. So these objective lenses are there. There are three objective lenses. You can like magnify as much as you can, and here this is for resolution to make the image clear. Light microscope is we say them because it is light is used here. Here is a mirror, and that reflects the light, and it goes through this specimen and into the or this tube and into the eyepiece, and we can see and observe the what type of object is placed there. So here there is another form of compound microscope. Here light is also used, but here this is a light bulb, which is enlightened by putting the plug in, and then this light is passed through this all object stage where the object is placed from objective lens, and this is eyepiece. So we can observe it clearly. This is the older one, just two lenses are there, and just this is a tube. Okay, so the photograph which we take through the microscope, whether compound or light electron microscope, that is called micrograph. So light microscope, it is having the magnification almost 1500 times than their original size, and its resolving power is 0.2 micrometer. This is actually micrometer. This is micrometer. Okay. So 1 micrometer is equal to 1 by 1000 millimeter. So that is, if any object is smaller than this one, we cannot observe through the compound microscope. And the smallest bacteria which is having this type of resolution power, so we can observe them through microscope, but not smaller than this one. Okay, if you see on any type of catalog book or any research papers, this code is written over there, LM109X. Just this is for example. So L for light, M for microscope, and its magnification is 109 times. So it means the micrograph is taken from compound microscope. Here we use now see electron microscope that is used in mostly in the research labs or sometimes in hospitals as well. So here is object lens also in this side, and here is the eyepiece. Two lenses are there, but there is a vacuum chamber. So vacuum chamber is used to pass electron beam to the object which is placed here in this chamber. So its resolution is 0.2 nanometer. In compound microscope, that is 0.2 micrometer, but that is very smaller indeed. And 1 nanometer is equal to 1 by 1000,000 millimeter. Its magnification is 250,000 times higher as compared to compound microscope. So its magnification was very less, that is 1500 times as compared to its original size. So this is more better, but there is some problem that when we see through this electron microscope, we can observe live processes, because live processes just have to be done in the air. So here is no air, there is a vacuum chamber, so live processes are impossible. For example, movement of any type of microorganisms, especially bacteria and other organisms also. So we must go to compound microscope. So now we'll see here transmission electron microscope. This is one of the type of electron microscope, and the other is SEM. T stands for transmission. Transmission means electrons are transmitted through the specimen. It means it passes through the specimen, any type of object, any type of cell. So we are able to observe all the inner parts of the cells. Inner parts of the cells in organelles, or like in nucleus, this is Golgi apparatus here, endoplasmic reticulum, vacuoles, and all the you can observe all the uh, organelles into from the cell. If we just see electron microscope, the other type, that is scanning electron microscope scanning, it just scans. It means the electrons reflected through the surface. So when we reflect it, just it means we can see just only surface, cell surfaces. It's uh, like cell wall and cell membrane, not inside that, like you can see here in this image. So this is scanning electron microscope, and computers are attached with this to see the image clearly. If we just talk about history of formulation of cell theory, very very important topic indeed, like how the cells are discovered. So that is very old, observations. So in the history of biology, if we see, ancient Greeks were the first to organize the data of natural world, like organisms are there, plants, animals, insects, birds, etc., and they've collected some observations from there. Aristotle, he gave the idea that all animals and plants are somehow related. So like all are having cells, they are having life processes, breathing, growing, etc. So the first uh, main discovery was given by the British scientist Robert Hooke in 1665, who discovered the cell first. He made his own microscope, and through this microscope, he observed a cork or piece of wood. Cork is actually the stopper of a bottle which was used in older times, and here this cork, because this is made up of wood, so wood comes from the plant. So when he see this cork is under the microscope, he saw some compartments like that, some holes in that, like the honeycomb. So he gave them the name celluli, and later the name has changed to cells. So the first discovery was given by Robert Hooke, uh, which is the discovery of cell. So he was not only just biologist, chemist, mathematician, and physicist. So he used some techniques, engineering techniques. So he made lots of devices like timepieces, quadrants, and Gregorian telescope. So the first discovery is referred towards Robert Hooke. The next one is very, very important also. Antony van Leeuwenhoek, he was a Dutch naturalist, and um, the first living cells. He observed actually the first living cells under the microscope, and he observed that there were the tiny organisms which are moving here and there, and then he gave them the name animalcules. So later this is called as animal cells. So like he just observed some microorganisms under the microscope or any type of lens, so he observed said them animalcules, and that is the specimen was taken through the pond water. The pond water means the dirty water where all the organisms are living over there. Another very important discovery goes towards Jean-Baptiste de Lamarck. So he was also the naturalist, and in 1809 he proposed that nobody can have life if its parts are not cellular tissues, because till that the cells have been discovered. So he was observing some more details about that. So he said to be that if the organisms are not having cellular tissues or cells, so they cannot be the living organisms. So later on, the discovery goes to Robert Brown. That is also very important discovery in the history of cell theory. That was the discovery of nucleus. Yes, he observed in 1831, because he was a botanist, so he discovered plants, and he saw just some dark spots in the cell, so he gave them name nucleus, because that is the central part of the cell, which does lots of functions. The next discovery was given by Matthias Schleden in 1838. So he gave that he was the German's botanist, is also botanist, and the first statement of cell theory. The cell theory is having three statements. The first statement was given by Schleiden, and he stated that all plants are aggregates of individual cells which are fully independent. Like all plants are made up of cells. So individual cells, when they make tissues and organ systems, then there is they form the living organism. This is the plant cell, and this is Matthew Schleiden has discovered this the postulate or the first feature of cell theory. Now, next discovery goes to Theodor Schwann. Um, 1 year later, he was because zoologist, like he studied animals. So in 1839, he reported that all animal tissues are composed of individual cells, like one cell, the other cell, the other cell, they form tissues and compounds. So altogether, Schleiden and Schwann proposed a cell theory in its initial form. So he proposed that all the animals are composed of cells. Schleiden proposed that all the plants are composed of cells, and Sean just discovered that all the animals are composed of cells. So later, Rudolf Virchow gave the next postulate of cell theory that is also very, very important. That he proposed that all living cells arise from pre-existing cells. Before this hypothesis, this postulate, the scientists believe that the like the life originated spontaneously, like itself. The cells originate itself, but like he gave this idea that the cells arise from pre-existing cells, like the cells which are before they divide and they are changed, like they divide and they form new cells. So that is called hominis cellulae, a cellulae. So that is a very, very important discovery, and that was proven. Like you can see here that the cells are dividing into two. One cell is dividing into two, and then they divide in the next cells. So they form the new cells. Yes, the next and this last postulate was proven experimentally by Louis Pasteur in 1862. He provided experimental proof of this idea, and in long neck flasks, he did some experiments about the discovery that cells originate from the pre-existing cells and not spontaneous origination. So these were some discoveries, and till the whole cell theory was discovered in its original form. So you can see here cell theory in its original form, now in its developed form. Okay, so if we see that all organisms are composed of one or more cells, now we know that all organisms are composed of one or more cells. These are some types of cells: egg cells, sperm cells, neuron cells, smooth muscle cells, bone cells, etc. So these are the cells, all the cells, just they combine, they form tissues, organs, organ systems, and they form an organism. So these all living organisms are composed of one or more cells. If one, they are called unicellular; if more, they are called multicellular, definitely. So next postulate is cells are very smallest or the smallest living things, basic unit of organization of all organisms. As the scientists discovered before, the living organisms, they are not organisms if they don't have the cells and tissues. So cells are the basic unit of organization, like they organize the body, and a functional unit also because they do the function of the body. And the next one, cells arise only by divisions in pre-existing cells, as we have seen in the previous slide, that there is one cell, this one like this, that it divides into two separate cells. So one cell divides into two cells, and 2 into 4, 4 into 8, and like that multiplication. Then it forms the tissues and complete a complete organism. So, dear students, that's it for today. This is very, very important. You must learn these three postulates, these three features, basics of cell theory on your fingertips, and you have to learn it properly, understand it. I hope you will find it interesting also. And if we see the history, you can see but through this timeline or history of the cell theory, by like how different scientists have discovered the cells and some of the postulates of cell theory. So that is like you can see the brief timeline of cell theory. So I think so that is enough. I wish you best of luck. Thank you so much for being in the class, and just find it very, very interesting and informative. Wish you all the best. We'll see you in the next lecture. Allah Hafiz.